My name is Marie Csete, and I'm the CEO of ConeSight Therapeutics. Obviously, not a lot of Hungarians in the audience. We are a cell-based therapy company dedicated to preserving and restoring vision. And in fact, today's talk represents our coming out of stealth mode. We're probably the earliest phase company presenting at this meeting, but I hope to get you very excited about what we're doing. So I was very well set up by the beautiful talk by Sue Washer. There's a huge unmet need in inherited retinal diseases as well as in complex diseases of aging, age-related macular degeneration. These inherited retinal diseases really have no therapeutic options at this time. We feel that our core technology, a cone progenitor cell, is going to be a game changer in this field. And we also feel that cone replacement is not only required for the inherited retinal diseases, but is essential for macular degeneration treatment in the patients with central vision loss. Our cone progenitor cell is a first-in-class replacement therapy. For those of you in stem cell biology, you know that generation of photoreceptors, cones, and rods has been a very hard problem, and there are no cone therapies in clinical trial. We're initiating a Series A funding round for $13 million that will get us to our first clinical trial in cone and cone rod dystrophies. Our cells are derived from a fetal source, and they are generated through a series of sorts and expands. And it is an extremely scalable manufacturing process so that one donor can generate sufficient cells for us to go through 2 phase 1 and phase 2 clinical trials and all the preclinical work that comes before it. And that's a very conservative estimate because we've not been able to demonstrate proliferative senescence of these cells, and we also think that our dose going in is less than the anticipated doses used in this calculation. We have a very good candidate identity marker. Purity using that identity set is greater than 90%, and viability greater than 80% at the end of the manufacturing process, both above FDA standards. We will show you some preclinical data that suggests that we not only have neuroprotection, but some true regeneration going on with restoration of visual function in several animal models. We haven't had a safety signal in our preclinical studies, and these cells are not pluripotent, so there's not a risk of teratoma formation. Importantly, what you need to understand about the uniqueness of this cell is that it's in a developmental sweet spot. So it's primitive enough, stem-like enough, that it just proliferates like mad. But it's committed to the cone fate enough that it only makes cones after transplantation, and they actually react to light in a dish. Our first clinical target will be cone and cone rod dystrophies because this is a huge unmet need. It's the tiniest part of the inherited retinal disease market, and the disease is extremely well matched to the product that we've developed. So cones are really important. We, as humans, depend on them much more for our daily functioning than we do on rods because they mediate your high acuity vision, your central vision, and your color vision. And as you can see in this diagram, the retinal pigment epithelial cells are the cells that support the rods and cones in the eye, and loss of both of them is characteristic of macular degeneration. And the cartoons that you see with the central vision changes are really cartoons, and they're not quite accurate, but they're a reminder of how important central vision is to our functioning. So in the first studies that were done at our founder's lab, Michael Young at Harvard, he did a retinal detachment model in the Long-Evans rat, and the first observation was just that there were abundant cells found in the outer nuclear layer a week after transplant. And Can I go back? Yes. In the bottom panel, you see the circled areas where the cells are labeled green as a human marker and red as a cone marker, and they're just easily found. And this is in contrast to the most relevant precedent in the literature and in clinical trials, the human retinal progenitors that were also developed in Michael Young's lab and then taken into the clinic by Reneuron. Those cells are doing quite well, but they do not make cones. And in the paper picture that you see at the right, great effort was made to find a human cone after transplantation in their preclinical models. So we were able to actually characterize this efficiency of engraftment by using the RD1, a retinal degeneration model in the mouse, where you can remove the retina after the transplant and splay it all out and account for every single cell in the retina. In a program developed by the graduate student in the lab, Pierre Dromel, we were able to count the number of human cells in the retina of these animals, and they accounted for 85% of the cells that we put in, at minimum, in each of these grafts. The other interesting thing about the way they engraft is that they didn't sit right next to each other. They engraft in a mosaic pattern, and that's how cones are patterned in the retina just outside of the fovea. So if you look at the lower right, that's an adaptive optics image of a normal human retina, and the bright cells are the cones arrayed in that mosaic pattern that you see in our animal models. One of the criticisms of cell therapies in this area is that you may be just seeing cell fusion to host cells that are remnant in the retina, and these were experiments done to show that that's not happening. So we, again, cut through the area of the transplanted retina in the RD1 model and labeled for human cytoplasm markers and labeled for human nuclear markers, and there was complete overlap of these. There was no cytoplasm in a mouse cell, and there weren't binucleated cells in the graft. So what about functional results? So unfortunately, the picture got very skewed on this, so you cannot see it, and I'll just refer to the graft. So we know that we have photoreceptor protection, neuroprotection, if we deliver the cells early in the disease, in this case, in the RCS rat model. And you can document and quantify the photoreceptor protection by counting the number of nuclei in the outer nuclear layer where photoreceptor nuclei reside. And at 50,000 cells, we saw that we had significant increase in the thickness of the outer nuclear layer. At 100,000 cells, we had even greater thickness of the outer nuclear layer. So at best, the cells are just neuroprotective, but we're hoping that it's more than that. The other way to monitor visual function after transplantation is by Electro Retinograms, and there's two sets of data here. Again, in the RCS rat studies, these were conducted at Oregon Health Sciences for us. The ERG is like the EKG for the eye. We saw an increase in the B wave, the upstroke, which is an indicator that we are perhaps activating the light transfer signal transduction to the bipolar cells, the next line of cells towards the brain. We also saw an increase in the A wave dip, which is a general marker of photoreceptor health. And on the right, ERGs were also done in the RD1 mouse, and the data is displayed in a different way. So an increase in B wave voltage was shown at both doses, though we found that the higher dose of 150,000 cells was too much. And it's really nice to identify a threshold dose. And in these mice, whose rods are all gone at the time of transplant, the ERG was only a cone ERG. We could not see a rod ERG, and that makes sense because the cells generating the electrical signal are the cells that we put in. The other way to demonstrate improved visual function is to stick a rodent in a rotating barrel that's striped, and they will follow the stripes with their eyes as their vision improves. And again, in the RCS rat model this time, we showed that 100,000 cells was better than 50,000 cells, and that 150,000 cells were too high a dose, even though there was improvement in the visual function at that dose. So this really supports the need for careful dose-finding studies, which we plan to do in St. Kitts in non-human primates. So if you look at the clinical trial landscape for cone diseases now, there are only 35 clinical trials listed on clinicaltrials.gov. The only cell therapy is Astellas' RPE therapy, not a cone therapy, for Stargardt disease. There are a variety of wonderful gene therapy approaches, some of which you heard about just now. The retinal implant is also being applied to cone disease, and new and repurposed drugs. We think we have an advantage over gene therapies in that our therapy is gene agnostic. And we also think that RPE transplants, though wonderful and safe, are not going to be the final answer without cones. Long term, most importantly, as other therapies come online for inherited retinal diseases and for macular degeneration, we think that these cones are very compatible with a lot of the anticipated therapies. Another important issue is that our therapy is useful at all stages of disease. So we show that we have neuroprotection early on, but late in the disease, when there's no cones around, cone transplantation is going to be one of the only options. Our regulatory path is underway. We had an Interact meeting in July, and it was a very positive meeting for us because we really reached agreement with the FDA on all of the issues that they brought up in their written response to our documents. So we have an ambitious course to move forward, and we're hoping that we will get to our phase 1 clinical trial in about 2 years at the Doheny Eye Institute in their beautiful new campus in Pasadena. Here is our breakdown of the costs needed to get to that first patient trial, and I'm going to point you to the bottom where I say for 2 clinical trials, because we plan on entering with the cone and cone rod dystrophies because of this perfect match to the product. But the preclinical package that we developed is also pertinent to the rod dystrophies like retinitis pigmentosa, so we hope to rapidly expand after our first patients are injected. And we're also looking for partnerships to start building a science pipeline for a macular degeneration program. And those discussions are underway with several companies. So we're really small. And we're building up now. Michael Young, the founder, also discovered the human retinal progenitor that I said was developed by Reneuron. And he is the discoverer, along with Deepti Singh and Pierre Dromel at Harvard, of this cone progenitor cell. I've been in this business for a long time. My clinical life was spent in solid organ transplantation, which I think is the perfect background for thinking about cell transplantation. And I've had the privilege of working with the Macular Degeneration Research Initiative run out of Doheny Eye Institute and the National Academy of Sciences for the last 13 years, which makes me realize that we have an incredible product on our hands. So I will summarize by saying our IP portfolio is licensed out of Harvard, but we're anxious to expand our IP portfolio in partnerships with a target towards macular degeneration, and I hope by next year we will have reported our successful open IND. Thank you for your attention.